Yeah, good day, students. So today this video is talk about the chapter 2, structure of cells and organelles. So the first thing we talk about the nucleus first. Okay. So first of all, we can see the cellular components. A typical eukaryotic cell has at least 4 major structural features: nucleus, cytosol, and membrane-bound organelles, and also the plasma membrane. Nucleus, found in all eukaryotic cells, which is a double membrane organelles, and they are not found in the mature erythrocyte, which is a red blood cell, and also the phloem sieve tubes. And then normally they have only one nucleus in each cell, except in the paramecium and the pseudocytic hyphae of some fungi. In skeletal muscle cells also have more than one nuclei, and we know it as a multinucleated cells. Anyway, today we just focus on nucleus. The function of a nucleus acts as the center to control cell activities and cell divisions. Because the nucleus contains genetic information, which is the DNA in chromosomes. Then the DNA can replicate and followed by nuclear divisions to produce identical daughter cells. This one that you have studied in the mitosis. The DNA also used to produce mRNA, messenger RNA, which is to be translated into proteins. And the nucleus functions also used to produce ribosome, RNA for proteins, and enzyme synthesis. So this is an anatomy of a nucleus. What you can see here is this is a eukaryotic cells. Then this is the nucleus. So in the nucleus, there is a densest area or the regions which is known as nucleus. Okay. Then the nucleus is enclosed by the nuclear envelope, which is a double membrane, and they have the nuclear pore, which allows the exchange of materials. Then you can see there are many chromatins. Okay, the chromatins are found in the nucleus. So for the nuclear membrane, you see it consists of the phospholipid bilayer. So they got two double membrane here. Okay, the nucleus is the largest cell organelle, as you know, and it's surrounded by nuclear envelope, okay, which is the double membrane. And there are nuclear pores in the nuclear envelope, and the space between the two membranes or the nuclear membrane is known as a perinucleus. Okay. Then the regions within the nucleus is known as the nucleoplasm. The outer membrane of the nucleus, normally they are continuous with endoplasmic reticulum and attached with the ribosome, which is the RER, the rough endoplasmic reticulum. This is for the outer nuclear membrane. So this nuclear envelope is perforated by the numerous pores. We call the nuclear pore, as I told you just now. And then the function is to regulate the passage of materials between the nucleoplasm with the cytoplasm. So the materials that can be passed through these nuclear envelopes are ribosomal subunits, RNA, proteins, nucleotides, ATP, and some steroid hormones between nucleoplasms and cytoplasm. Okay. Inside the nucleus is the nucleoplasm, which contain the chromatin. You can see the chromatins. RNA normally found in the nucleus, and the nucleus, nucleotides, enzyme, and minerals ions. Then the chromatins consist of DNA strands associated with histone protein. Okay, when we compare to the prokaryotic DNA, which has no histone proteins. Then this DNA, they're coiled with the histone proteins and condensed to form a chromosome during cell division. So as you can see here, the chromatin is a long thread of the DNA. Okay, and then they associated with histone proteins. So during cell divisions, these chromatins undergoes replication, or we call it DNA replications, to form the sister chromatics. Then after they were condensed to form chromosomes. So this is what you learned during the Form 4 and Form 5. Okay, so this is the structures of the chromosome. You can see the chromosome here. If we pull the chromosome, the DNA out, you can see actually the DNA, they coil with the histone proteins. Okay, so this is the double helix of the DNA. Okay, so they coil with the histone proteins, then they further condense to form the chromosomes. Okay. All right, this is what you see. This one small clear because the diagram is bigger. This one is a chromosome. You can see this is what you have learned in the chapter 1, the DNA double strand. Then you can see they coil with the histone proteins. Then after that, they form the nucleosomes. Nucleosomes, they are the DNA they associated with the histone protein. We call it as a nucleosome. Then after that, they further condense and coil. We call it a supercoil structure. So after that, this one form a chromatin. And then after that, they further condense becomes a chromosomes. So under the microscope, two kinds of chromatins can be distinguished according to the state during the interphase. Okay, you can see the heterochromatin and euchromatins here. So heterochromatins, they are coiled, the DNA are coiled with the histone proteins. Euchromatins, whereas there is no further coil of the DNA. So what is the difference here? So the heterochromatin normally they are stain density and is generally not transcribed. That means they cannot be used to form or to produce mRNA. Okay, then the gene in the heterochromatids can only be transcribed if it uncoils to form an expanded chromatin, or we know as the euchromatins. Like here, this one is heterochromatins. Then after that, they uncoil becomes a euchromatins. So now here they can be the DNA or the genes can be transferred into an mRNA. So euchromatin is an uncoiled and stained lightly. Okay, and contains the DNA that is transcribed into the messenger RNA. So from here, euchromatins. Okay, like just so you can see the one. Okay, nucleus is stained darkly in nucleus. So this is the nucleus. You can see this dense darkly. Okay, the denser regions, which is known as the nucleus. It contains DNA. Okay, the DNA is here, and ribosomal RNA. Okay, the rRNA. Because the rRNA will be formed by the DNA in this region, and proteins, they are needed in the formation of ribosomes. The protein actually is synthesized outside. Okay, that means in the cytoplasm. Then after that, transported into the nucleus through the nuclear pore. And then these proteins will bind with the rRNA or ribosomal RNA to form the ribosomal subunit. Okay, as you know here, rRNA is synthesized in the nucleus. Okay, then the proteins needed to make ribosomes are synthesized in the cytoplasm and imported into the nucleus, as I explained to you just now. Then in the nucleus, the rRNA is folded and combined with the proteins to form the ribosomal subunit. If you study in the previous lesson, you already know the ribosome. A complete ribosome actually consists of the large ribosomal subunit and the small ribosomal subunits. So this large and or what we call it, this subunit, ribosomal subunits, they are formed in the nucleus. Then later they are exported or transported to the cytoplasm for complete assembly. Okay. During nuclear division, the nucleus seems to disappear because the DNA are dispersed. Okay, the DNA are dispersed to form the chromosome. So that's why the nucleus is disappeared. Then they will resemble after the nuclear divisions. So that's all for the nucleus. So I hope you can understand this about the nucleus. So if you have questions, please contact me. Okay, so that's all. Bye bye.